Hello and welcome to this video on a technological method used in microscopy called fluorescence microscopy. As the name might obviously tell you, the whole premise is to make something inside your cell, tissue sample or anything else glow under a certain light. And now we should be clear that this is typically used in biological sciences, but it can find roles in others where making fine details obvious is useful. Let's start with a simple example that should help to understand what and how this is useful. Let's say you have a cell. This cell is part of an experiment to understand how a certain thing acts. For example, a movement of a protein. And by adding a small marker to the protein in the form of a fluorophore, or something similar, means the thing that you're interested in will glow. Well, typically it will glow if it's added to the same place as the protein, since it is part of the protein now. Either that or it's become attached to it. Now you use a microscope made to apply a certain wavelength of light. That wavelength of light should correspond to the wavelength at which the thing itself will fluoresce, and so, with the fluorescence, you can now image it. This will allow you to see the thing you're interested in without having to look at all the other details necessarily. This is similar to how you can genetically modify an organism to be visible on the whole. This is typically done using green fluorescent protein and is a very common undergraduate experiment to demonstrate genetic modification. In other cases, it might be useful to use a liquid version of this, sort of like a paint that flows into things like gaps and cracks. Then you wipe away or otherwise remove the material outside the gap. You repeat the previous application of light, but the fluorescence lets you see where the cracks that the fluorescent compound leaches into much more easily. In other cases, you can use what we've done in the past. It can act as a tracer or indicator of the presence of something, such as with our homeopathy dilution factor video. It's important to note that not all chemicals, organisms, etc. require external sources of fluorescence, as they are autofluorescent or self-fluorescent. This means they're able to shine themselves so long as you put light into it that will stimulate this process. For the most part, this isn't an example of that. This nearly always involves an external and artificially generated fluorescent compound. In terms of those compounds, there are three major approaches, and they cover what we've described so far, those being stains, immunofluorescence, and proteins. With that, let's go through each. The stains are arguably the first and easiest to understand, as they are quite literally a case of having a liquid, just like with clothing where you apply it and it alters the appearance. Now, unlike most products, let's say, that are used to stain clothes or similar fabric, you don't want it to stain absolutely everything. Typically speaking, these are either used in a way where, to put it simply, you have a certain compound that allows it to be more permeable to certain parts of the cell or similar thing of interest to you. This means that it only builds up in certain areas. For example, a fairly common example is DAPI. DAPI or DAPI allows it to enter the nucleus, and therefore the stain is really only present in the nucleic acid area of the cell. That means you really only see where the DNA is, and to a certain extent RNA as well. This can be very useful if you're trying to trace something from, say, the nucleus to the RNA and see where the RNA is building up, if you're looking at, say, messenger RNA, for instance. Of course, this isn't the only time and way in which you can use it. In other instances, you may have, you can certainly look at trying to stain certain things within the cell. For instance, actin, and you can look at where the actin is at given stages of cell development. So, for example, during cell division, where you have actin fibers building up within it to help with division. There are also other ways to do this again, although these other ways, which we'll get to in a moment, overlap considerably with immunofluorescence. And that's because you typically have the fluorophore, which is what you're using to stain and therefore be able to see what's happening, attached to something else. Now, not always the target site. Sometimes it's an intermediary. Of course, it's not necessarily the case that you need an antibody. In some cases, you can use another intermediary compound, chemical, or structure to allow you to specifically bind to certain things within your cell or sample of interest. But for the most part, immunofluorescence, which is our next technique, is the mainstay of staining like this, and the name is more or less going to tell you exactly what's happening. First of all, you have your fluorescent thing, your fluorophore. This is a chemical that will respond to a certain wavelength, and it will emit a different wavelength, so you can see what it is you're looking at. Then you attach this to your antibody. Now the antibody is going to be specific to whatever it is you're trying to target. So for instance, if you're trying to find certain cells within a larger sample set, if you are able to take some of them and get the antibody to it, you're then able to generate this antibody, and as long as you can bind the fluorophore to it, you can then create this targeted and very specific fluorescence on only what you're after. This process of only looking at one antibody with the fluorophore directly attached is called a primary antibody. In many cases, this doesn't always work, and in some cases it's undesirable to have a primary antibody only. And this is particularly true if you're looking at something where you want to differentiate two very closely related things. This is where you may use a primary antibody and then a secondary antibody. The primary antibody doesn't have anything attached to it, but it's reactive to the secondary antibody that does have the fluorophore attached. A simple example would be, say, anti-whatever your target site is as the primary antibody, followed by a secondary anti or whatever it is that you've derived your first antibody from. So for example, anti-actin followed by an anti-mouse, because mouse is where you got your first antibody from. This means you can ensure specificity to your targeted site and thing of interest. This is because not only do you have to have the primary antibody bind to it, but the secondary antibody has to bind to that first antibody and often to a secondary thing of interest. This ensures that you only get let's say a result if both things occur. Finally, we have proteins. Now, proteins are actually perhaps the easiest and hardest thing to do, and that's for a simple reason. The easy part of it is down to genetics. We have genetic modification technology. This allows us to modify the DNA and allow for proteins to be able to carry the fluorescent protein reporter. That is the fluorescent compound. The challenge is ensuring that the fluorescent compound doesn't get in the way of the protein doing its job. When this works as intended, it allows for the protein itself to have an inherent fluorescence, and therefore you can locate and track proteins without any issue whatsoever, and without having to add anything at all to the cell. So you don't need stains, you don't need antibodies, you just are able to image the cell directly. This is ideal if you're trying to look at a live cell that's working as it's meant to be, rather than trying to catch cells at different stages and get snapshots of time and basically have a single destroyed sample at the end of that. This, of course, isn't necessarily perfect. For example, the issues around the whole structural integrity and function of the protein can be undermined by this. But if you find a way to make it work, it is arguably the best way to approach proteins. At the same time, it does, however, come with certain caveats that make it more difficult to achieve what you want, particularly around the time to develop exactly the right modifications. In fact, this is why these techniques, and particularly the end result, are not always the end goal of the research in and of themselves. For example, it may be part of proving that a certain protein is involved in a specific pathology, or the etiology of a certain pathology. A simple example is the movement of tau proteins in the brain and the presence only in those with Alzheimer's, or the degree of tau bundle formation and dementia symptoms. By itself, this is not enough to establish a model, and certainly not causality, but it is one part of the wider picture. So you might be looking at the end results of your staining. You might then look at trying to genetically modify an organism, wherein you know that your particular model organism will develop dementia, and you've modified the tau proteins such that they will have native fluorescence. This means that if you're able to show that in a pathological sample from someone who's died and donated their brain to science, and in your mouse model you both have dementia, you might be able to say there is a relationship. In other cases, this whole process might actually be the end goal, at least for now. A recent example of this you'll have seen running in the background is the egg embryo and actin protein. The embryo model was able to be genetically modified, and by modifying the actin protein, you're able to visualize the movement of actin protein in greater detail, and it even allows for real-time monitoring and recording of actin location and density. Despite how cool all this sounds, there's one more thing that needs to be discussed. Simply put, it's why fluorescence microscopy is not perfect. The first reason is simple enough. The fluorophores are just like glow-in-the-dark paint. They will lose their ability to fluoresce as they are eliminated. It's a process called photobleaching. Photobleaching occurs because the molecules accumulate chemical damage. Now, the chemical damage is a result of electrons being excited, which is exactly how you get fluorescence. It can also, for a certain argument, limit over time the ability to accurately and reliably image the particular sample you're looking at. You can gradually increase the amount of light you're introducing to try and compensate for this, but then you begin affecting image quality. So there's a point at which you get diminishing returns, and at that point you're really better off just giving up on that sample and moving on. The other issue is particularly around live cells. Now, we've mentioned that recent research, and understand this is recent as in only the last few weeks as it being published, that you can look at live cells and keep them alive the entire time. But when you do so, and one reason why they had to use bird embryos in egg, is that the process of exciting the fluorescent compounds can lead to phototoxicity. This is particularly true if whatever fluorophore you're using requires a relatively short wavelength, basically UV light, and as you would be aware, UV light, given what it does to the human body and skin cancer, has an equally if not greater negative effect at the scale of a cell, where there is no protection from layers of dead skin cells, let alone everything else that the human body can do in defense of, well, for the most part, sunburn. This means that the molecules, both through the process of excitation to get that emission of light, can cause damage, but the actual fluorescence itself, due to the nature of what it does, can generate reactive chemical products. Oftentimes it's reactive oxygen species, and this means they become even more toxic. And then finally we have, well, the issue around the technique itself. One of the major challenges, at least until relatively recently, and certainly in a more broader sense, yes, is that you can only look at specific things. And that's oftentimes because what you're trying to do is use one fluorophore, although you can sometimes use multiple, to stain a single structure. And when you have that single structure, you are only able to look at that alone. Now, if you add 2 or 3 stains, you might be able to look at a few more things at the same time if they have a similar excitation wavelength and very different emission wavelengths. But you're really limited in how much you can look at any given time. And certainly with less innate or intrinsic methods, for example with stains and immunofluorescence, you really can't look at a lot of different things at the same time. This means that you're really limited in how much you can examine, and typically speaking, it only tells you as much as what you're looking at, because the way the microscopes are designed for this, they really only let you visualize what you're looking at and looking for with terms of the fluorescence, and not necessarily anything else. So for instance, if you're looking at that nucleic acid staining method from earlier, you can tell where the DNA is, and you might be able to make it such that it also stains to RNA. So now you have DNA and RNA stained, but you don't really know what it's doing necessarily. There are of course ways around this, but the ways around this may not necessarily be useful in extrapolating out, as you're going to struggle to ensure that the sample you're looking at, particularly after the fluorescence process, is exactly how it should be. Other times, you basically have to accept that this is just one arm of an experiment, and you have to justify its reliability and context using other methods again. This is what fluorescence microscopy is. It's a process of creating highly detailed and specific images using either integral parts of the cell or organism, adding parts, or staining, but importantly, you're only looking at specific elements of anything you're trying to examine, because everything else doesn't really show up, and that's the challenge here. It's a brilliant technique and technology in isolation, but it's also somewhat isolated in its application in most instances, because it can only account for so much and only what it's able to image. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. But please do post any comments, questions, or suggestions you have below.